Well, welcome everybody. My name is Florence Hudson, and I'm Executive Director of the NSF and NIH-funded Northeast Big Data Innovation Hub at Columbia University and founder of the National Student Data Corps. I want to welcome you to the new National Student Data Corps Masterclass Series, which we're kicking off in August of 2023. The National Student Data Corps was created at the very end of 2020 and beginning of 2021 to provide an opportunity to bring awareness of data science and education to students, communities, and institutions around the world that are underserved from the perspective of having access to data science education resources. Either their school doesn't have a data science program, they're not in school right now, they don't have some of the advanced concepts, or whatever it is that would be a detriment for them to learning data science, because there's so much opportunity in data science careers. So we've been doing a panel series where we had career panels with various subject matter experts across many different domains: music, sports, statistics, bioinformatics. Now what we're doing is creating a masterclass series, and this is the first one, so welcome to the masterclass series, where we want to have experts like Ian, I'll tell you a little bit more about Ian in a minute, who actually can talk about how data science, artificial intelligence, machine learning, knowledge graphs are used for specific domains, as we say, data science for good. So in this first episode, we're going to have an introduction to AI and precision oncology. And our guest speaker today is Ian Maurer. Ian is CTO at GenomOncology, and you'll learn a lot more about him in a minute. And I want to thank Emily Rothenberg, the National Student Data Corps Program Coordinator, who put all of this together. So on our next slide, we'll see that our agenda today is an introduction to AI and precision oncology, which Ian will do. Then we're going to have a little Q&A discussion, and hopefully we're channeling some of the questions in your minds too. And then we will talk a little bit more about the Northeast Big Data Innovation Hub and the National Student Data Corps, so you can stay connected and take advantage of all the resources that we bring to you, open, online, and virtual around the world. So now what I'd like to do is pass it over to Ian. Ian is going to share his slides, which are really enthralling. He just gave us a little run through. And Ian, take it away.
Great. Thank you so much. Glad to be here. A little bit about my background. I have a master's degree in computer engineering from Syracuse University. I'm the chief technology officer of a company called GenomOncology, and I'm the AI working group co-lead for a group called the Cancer Informatics for Cancer Centers. GenomOncology is located in Cleveland, Ohio. We have 17 NCI designated cancer centers that we work with. And we also have an extensive knowledge graph that has clinical trials and therapies and approvals for cancer drugs. And that knowledge graph is what I'm going to talk about today in conjunction with artificial intelligence. So let's start with what is AI? What is artificial intelligence? It creates value through scalable probabilistic prediction. It's scalable because we can, rather than having a single person working on a single task at a time, we can scale out to tens and hundreds and thousands of computers to run prediction algorithms. These prediction algorithms are probabilistic. So unlike software code, which are deterministic and will give you the same answer every time if you ask for it, these models are probabilistic in that they could give you different answers for the same query. And that there's really not a way to know for sure it's giving you the quote unquote right answer. So systems will have to be redesigned and rethought of to actually take advantage of a lot of the value that these artificial intelligence capabilities produce. Some high level terminology, some lingo, because one of the major barriers to actually getting involved with AI is really just understanding what folks are talking about. And this shows the high level flow. And the key elements are really starting with a data set to build a model that you can then use for prediction. And the words folks use to actually say the process of building that model is training. So you're taking that data set, you're training a model on a given architecture, so then you can do inference or ask your questions to get your predictions back. And the architectures are various architectures. There are machine learning architectures, deep neural net architectures. GPT, for instance, is a type of architecture, generative predictive pre-training. And that is a particular type of architecture that they've trained basically in the text of the internet It's more complicated than that, but they've taken the text that's available on the internet, and they've trained a model to predict the next word in all the sentences in that text that they have. And then what is precision oncology? So precision oncology is really the concept of diagnosing and treating cancer patients through mutations. And those mutations can be either something patients inherit, what's called their germline, or that evolve in their body over time, and that's called somatic mutations. And the types of mutations that we find that lead to cancer or contribute to cancer are, first, there's oncogenes, where if they get mutated, they can result in uncontrolled cell growth. There's tumor suppressor genes, which do the job of trying to stop that from happening. And if they have mutations that kind of break down their capabilities, can lead to that, let that growth go unchecked. And then there's DNA repair genes. As cells divide and the DNA divides, errors emerge just based on the copying mechanisms. These repair genes are responsible for correcting those errors. And if they fail to do their job, it can lead to higher prevalence of cancer. And then in precision oncology, the goal is really to come up with better treatments that have less side effects for patients. And there's really two emerging types of treatments. There's targeted therapies that attack the cancer cells directly, and there are immunotherapies that boost our body's natural defenses. And this field has really started to take off in the mid-2000s with the invention of next generation sequencing. And you can see here, Moore's law is a well-known definition of why computers have gotten bigger, better, and cheaper over time. It's really just the cost of the transistors and the cost of creating CPUs and GPUs. The cost of per genome getting analyzed has gone down precipitously over the last 15 or so years. And that's really because of this next generation sequencing. And this has really unlocked this capability to let us treat patients, cancer patients, in a more cost affordable way. And it's not just next generation sequencing and genomics. There's really multiple omics that are involved. And omics is the comprehensive study of biological systems. And it includes genomics, which is the analysis of the DNA. But there's also transcriptomics, which analyzes the RNA. And then there's proteomics that analyze the protein expression. And so all three of those are available in use today in the clinical setting. And we see IHC, or immunohistochemistry, and next generation sequencing, NGS, quite often with our clients that are developing tests for specific biomarkers. And the challenge and the opportunity is the fact that there's just a lot of available treatments over the last 10 years or so. And that has also led to more clinical trials as well. And so, drug companies are coming up with new treatments. They're testing those using clinical trials. They're looking for patients that have specific biomarkers, and recruiting for those trials is a challenge because it's really a matchmaking service. And that's one of the things our software can do. And then the other challenge is keeping track of all of the available therapies and making sure that doctors are aware of the best possible treatment for their patients. And that's where GenomOncology comes in. So our first product was what we call our pathology workbench. And what this tool does is it basically takes data directly off the sequencer. The data comes out in formats. There's a file format called a VCF, which has the specific variants that were found, and then other information as well that's interesting to look at from a quality perspective. Our system is really designed to be a high process workflow so that the pathologist and their team can review the information, the data quickly, get comfortable with what the variants that were found are, and then interpret those variants, say which ones are actually pathogenic or benign. And the pathogenic ones are those oncogenes and tumor suppressor genes that I talked about earlier. And then the goal of that process is to then generate a report that can then be sent to the treating oncologist so that they can help their patient with the most information. And that report has on it the specific variants that are of interest, the specific trials and therapies. And this is just an example of our sample report that we create. You can see here that there's significant genomic variants that have been detected that are actionable or not actionable, and hopefully that will be future clinical trials for them. The pertinent negatives, because a lot of drugs and trials depend on knowing that a patient doesn't have a specific type of mutation. And then the actionability. So this is, some information that includes maybe FDA and NCCN guidelines. We have guidelines from the UK and Europe as well. And we're constantly adding new information to our knowledge graph as we expand to other markets internationally. And so how does it all work? Well, the first thing we have to actually understand is the biomarkers, right? And like I said earlier, it's multi-omic, so it's not just NGS. There's IHC and FISH and cytogenetics, lots of different ways of detecting and finding biomarkers. We get data both in structured and unstructured format. For unstructured data, we actually do use AI, a subfield of that called natural language processing, to extract data out of unstructured data formats. And we convert it all to standards. So there are a variety of standards like HGBS and ISCN that we convert the data to so that we can actually use it in our matching algorithm. And, over here, you'll see that there's specific mutations that are detected for patients. And they basically are telling us what chromosome did the mutation happen, where on that chromosome did it happen, and what was the change. What was the reference, meaning the expected value, and what was the actual change that happened. And in this case, there's a change of 3 base pairs being deleted from a gene called EGFR. And then in our database, we have a variety of curated concepts that are meaningful in precision oncology, whether there's a trial or a therapy, or just means that the variant is quote unquote pathogenic and therefore needs to be investigated more. And then our system can actually link these two together and unlock features like matching to trials, but then also doing things like cohort analysis. So, for researchers, there's lots of times where you have to go find a cohort or group of patients based on some set of clinical information or biomarker information, and our software helps folks do that type of research. And then another key thing our software does is a variant interpretation engine. And this is a version of a decision tree, right, or old school, artificial intelligence system. And what it allows our molecular pathologists to do is to analyze and interpret a variant quickly and give them the exact reason why the system made that recommendation. So what happens is variants come into the system, and if they're not known to be benign or pathogenic, our system can walk that variant through an interpretation template that's totally configurable and understandable by our clients. And then it gives an interpretation result and says this variant is probably pathogenic, and here are the reasons. And you can see over here kind of a blurred out version of what that tree might look like for a client. And every client has the ability to kind of configure and customize this capability in our system based on their validation of their test and some of the other operating principles of that lab. And then once we've interpreted the variants, we can then use those interpreted detected variants to then see if there's match from a content perspective. So that content could be prognosis, could be risk, it could be a clinical trial, or it could be a therapy. But we have a team of folks with PhD background in genomics and cancer that are reading the literature and the clinical trial eligibility criteria, and they're curating rules within our system. And these rules take a shape of a nested Boolean data tree. And effectively what it says is, in this case, it's saying a patient can qualify for this rule, which happens to be a prognostic rule by NCCN, or an old one. And it says, if you have either a frame shift or a nonsense mutation in this gene called ASXL1, those are types of mutations, and you have a normal karyotype, meaning your chromosomes look okay underneath a cytogenetics microscope, and you have none of these other mutations. If all 3 branches of this tree are true, then you qualify for true at the top, and then you'd be able to match to this particular rule, and it would move on to the next stage of our algorithm. And this is what our whole high-level algorithm looks like, right? We take a patient, their age, gender, zip code, all of their multi-omic lab results, because we want to look at all the variants at once, not just a specific one at a time. And then we look at prior interventions, because some therapies and some trials preclude folks that have had a specific type of drug in their background. So you need to know that information as well. We can then interpret the variants, match the patient to the right trials and therapies, and then we provide that information in the context of our facilities, so they can actually filter and augment that information with maybe what clinical trials are available at that institution and that are actively recruiting at that time. And then we generate a set of recommendations. So our system is called decision support. We don't make decisions. We just surface high quality information to treating physicians, pathologists to make their decisions. And that can include things like, what's the interpretation of the variant, what are the trials, and then clinical pathways, which are basically decision trees that physicians will go through to try to find the best option for a patient at a given time of their treatment history. And we support a variety of different workflows like reporting and what are called molecular tumor boards and alerting, and then analytics platform as well that allows folks to make charts and graphs and do cohort analysis on patients for drug discovery purposes or clinical trial recruiting purposes. So now, what about large language models like ChatGPT? The world has changed quite a bit since last fall. This was a story in the New York Times about a lawyer that used ChatGPT to basically write their legal brief, and basically it included citations in it to cases, and those cases didn't exist. So when you're using these tools, you've got to recognize the fact that it's not a database. It's not going to give you cited information, and it can make things up, which are called hallucinations. And why do they hallucinate? Well, first, large language models struggle with queries that are ambiguous. So a lot of times folks complain that they're getting the wrong answer out of a large language model. Well, sometimes you might want to look at how you're asking your question to make sure you're asking it in a clear and unambiguous way. But another big challenge is the large language models, when they're uncertain, they don't communicate that uncertainty. They'll just talk with authority and when giving you their answer, whether they actually, because it doesn't actually know the answer, it is just predicting what the next word would be to a good sounding answer. And then there's a limit to the data set, right? So like I said, it's trained on the words on the internet, but it's not trained on all the databases in the world, like genomics databases and other very comprehensive data sets. It's out of date. So training these systems is very expensive, and updating them is not easy. So they get out of date based on when they were pre-trained. There are tools that we can use to incorporate, latest information. I'm going to talk more about that in a moment. And then they can't really verify and source their attributions. They can't provide citations because it's not a database. It doesn't have it. And if you ask it for a citation, it will give you one. And if it's to a well-known website like Wikipedia, chances are it's right, but everything else would be very suspect. And you would have to click the link, make sure it's actually there, and use it. It also has limited working memory. So there's only so many words that it can actually keep in its memory at one time. And so you'll notice if you work with ChatGPT a lot, you'll find that it actually starts to lose the thread of the conversation and forget earlier things you told it, or even earlier things that it has said. And so our way of combating that in our field is to use our evidence-based knowledge graph, right? So we have this knowledge graph. It's got fundamental building blocks, or what we call ontologies. Start with genes and transcripts and diseases. Those building blocks, along with the annotation sets around these variants, and this is more around the information around what's been published, what are some of the prevalence, how much does a particular variant happen in the public. And then we use those two items to then build up our assertion model. And the assertions are basically statements of fact at an intersection of a set of concepts. So a clinical trial or a therapy is a statement of a patient's eligibility based on the concepts that I talked about earlier in that Boolean logic tree with the any, alls, and nones. So our team is curating this list of assertions continuously, making sure it's up to date with the latest FDA approvals, the latest clinical trials. And really at the end, what we have is a good old-fashioned symbolic AI system that allows you to do reasoning, but also is a white box where you actually understand and can verify why specific recommendations were made. It's including, who on our team actually made a creation of a assertion in our system. And then combining these two things together, large language models with knowledge graphs, really give us the best of both worlds. In this book by Daniel Kahneman called Thinking Fast and Slow, he describes system 1 and system 2 as being a way that a human mind might actually work. Where system 1 is the pattern recognition, the automatic and unconscious decisions that we make when we're driving or some other activity. while system 2 is the slower, step-by-step planning and deliberative thinking that we do. And really those two systems together allow us to make, good decisions, but also to react quickly to our environment. And I see large language models paired with something like a knowledge graph as a way of getting the best of both worlds, being able to interact with people in a language way, getting, understanding questions, making the queries against a known database, getting those results, and then communicating those results back in an audience-specific manner. And then I have a full demo of this on YouTube that you can search for, called Grounding Medical Q&A using ChatGPT plugins. And in that, I talk about this exact concept using OpenAI's plugin capability, where it's calling my API to answer specific questions around the latest drugs that are available and the latest clinical trials that are available for patients. And please don't hesitate to reach out to me. I'm on Twitter and LinkedIn, and there's my email as well. And thank you very much.
Ian, this was fascinating, and I think our listeners are going to be really enthralled with not only hearing how artificial intelligence and decision support and knowledge graphs models are not just something you learn in school or learn on their own, but how they can apply to things like precision oncology, where you can gather new insights and knowledge to be able to better care for cancer patients. That's amazing.
Great. Thank you.
And I really like how you explain that the system is a decision support system. It's not a decision-making system.
Right, exactly. Very, very, very big caveat for our company, and if the FDA is listening, for sure.
Oh, absolutely. And
Yeah.
and so the system provides these knowledge and insights to humans who are responsible for the actual decisions. And I'd be interested in you all at GenomOncology have deployed it for this very specific purpose. But when I think of ChatGPT and the way people can use it, like, we at the Northeast Big Data Hub and the National Student Data Corps and the COVID Information Commons, we have students that write papers and do posters and stuff. And we say, you can use that stuff, but you're responsible. You are responsible for the output,
Right.
just like what happened with that lawyer. Is that how you see it? Is that
yeah, for sure. For sure. Yeah, and like 6 years ago, the chairman of my company was like, all right, we got to get on this AI bandwagon, effectively is what he said. And I tried to explain to him, because I was very anti the word AI just because of the baggage involved with it. So I really pushed the idea of augmented intelligence, right? What we're doing is we're not trying to replace people. We're trying to make people better and faster, right? So Steve Jobs has the bicycle of the mind video on YouTube, if you've ever seen that. And that's kind of the analogy I go with. And, another one I'll say is amplified expertise, right? Basically, people have expertise. You don't need to have everything at your fingertips. Like, you don't need to remember everything, right? So being able to have these tools will let you focus on your judgment and let you focus on, decision making and dealing with the uncertainty and bringing your human values to the situation. And our goal is never to try to replace doctors and these companies that are out there saying, oh, we're going to get rid of all the radiology jobs. It's ludicrous thing, right? Like you're basically what's the word I'm looking for? You're turning off your main buyers, right? The people that you're trying to replace are the folks that are actually going to buy your system.
Yeah, and you would never do that, as you say.
Yeah.
It's, I look at it, I worked on AI and deep QA and deep learning years ago at IBM during my career there. I worked on the Watson strategy. And we always thought about it, as you said, as augmenting a human knowledge, not intelligence. Like the human is the intelligent one, like they're, and it's really augmenting their knowledge because there's so much going on out there on a day-to-day basis, new research, new, clinical results. And doctors are brilliant, but they don't have time, to comb all these resources every day. And so with your system, how often is it using like new information that's available out there from research or from, clinical results or things that you would find in the National Library of Medicine or whatever, versus it, being, kind of a static database, so to speak?
Yeah, so there's two answers to that. First is it's updated every day, really, and we deploy it to our clients every week if they're able to accept data updates, because everything we do is installed behind their firewall and on premise. So we can deploy our data and our code every week to folks that can take it. And that means it has the latest trials, the latest FDA approvals, right? So if there's a special status approval that came out, our doctors are going to want to be able to see that and have that on their report for timeliness purposes. The other question really becomes around what we call lower levels of evidence, which in the clinical setting has less, I won't say less value, I guess is a good way to describe it, but it's not as important in the clinical setting, just because the latest research is interesting, but what you really want to be able to do is help a patient with a treatment or a trial, ideally. So what we've done is we've partnered with a group called the Jackson Laboratories, and they actually do lower levels of evidence. So they're keeping on top of all the published journals that are coming out, and we have all those variants in our system as well. So when you get the report, and the report's 15 pages, the last few pages will be about, hey, here's some addendum information about these variants, and where you can find more information about them, but they're not clinically relevant at this time.
That's really excellent, the way you kind of parse the information to make it clear, like, think about this, consider this, look at this,
Right.
some new pertinent information for you. And I think I kind of think of these systems as kind of like data Sherpas in a way, as we get into this very intense, connected and data rich world where you're not sure of the provenance of the data. There are a lot, like where it came from, can you trust it? So I think there's so much value in companies like this, that can do that and help the experts, the doctors in this case, to be able to leverage the information, but they can still choose what information they want to use, right? Like, can they look at where did this, data come from, where this information?
Correct. They're making all the decisions, right? So our system, for instance, so we'll give them all the options of trials and therapies. They can deselect anything they want. They can say, oh, let's not put this on the report for whatever reason. And the same thing happens with the variant interpretation. So meaning, we get a new variant, we haven't seen it before. It's called a variant of unknown significance, or VUS. Our system gets that, evaluates it through that decision tree, and then makes a recommendation. That doctor at that time can say, nope, for this case, I don't agree with that interpretation. I want to change it based on something I know. And I'm going to put a note in there and save it. And then in the future, someone else could, when they're looking at it, the same VUS, they can either choose the automatic thing, or they can choose the doctor's previous this override. And those overrides could also be more case agnostic, right? Where you could say, okay, in this, for this variant, for this reason, because there's a paper or whatever that's not in some annotation database, I'm going to say that this variant is pathogenic, even though the system's saying it's benign. And I can cite my resource and then hit save, and then it'll actually affect all of the future cases as well. But at the same point, once again, it can always be overridden, right? New information comes out, let's go ahead and override that. And then, this constant battle of, just keeping on top of the latest literature and the latest options. That's a key part of what our system does. And what you want to be able to do is at any point in time, look back at a case and say, for this patient, why did we make this recommendation 2 years ago? Well, you need to know what the data was and the knowledge was at that point in time, because that's a critical component to this concept.
Absolutely. And that's just so important, that what you're continually saying is it's the human intelligence that's the most important, and we're just augmenting it, as you said. I totally agree with you. One more precision oncology question, in precision medicine, and then I'd like you to share a little bit more about the paths that some of our listeners can take to get into a company like this, or into this type of role, either yours or the PhDs you talked about. So in the work that you're doing in the precision oncology, is the precise care recommendation always for a particular human, or can it apply to a broader cohort, with a similar type of biomarker or other types of similarities?
Yeah, it can be both, right? So, and it really just comes down to, false positives and false negatives and what your goal is, right? So, okay, first, I've got to, let me bring up that one slide for match. This slide will help explain it.
Yeah, I thought it was both, but I just wanted to ask so that people could hear, the broader impact, as we say in NSF land, of what you're doing. It could be for one human or it could be for a cohort.
Correct. Yeah. So what we're doing in Match is given all the information we can get about a patient, because the more information we have, the more false positives we can eliminate. So what our system tries to do is have no false negatives, meaning we don't want to miss something, right? We want to present all the potential options, allow the doctor to filter things out, sort things however they wish, and bring their decision to it. The worst case scenario for our system, from my perspective, would be we didn't provide an option that was available. So those are false negatives, and we do our best to never have them. The problem with that is now you get overwhelmed with options, right? Clinical trials. I was just talking to an MD yesterday, and she said, I had 5 trials that were available. Even that exercise of kind of going through the trials took 2 or 3 hours just to try to find a good match for my patient. And so whatever we can do to remove false positives, and the false positives could be that trial's no longer recruiting because there's some updated status in the database, or maybe there was a prior treatment that needed to be categorized, or there's some other element to that specific patient that would eliminate that as an option. And the more we can eliminate options that are not valid, that's better. So the inverse of match is the cohort analysis. And this idea is I have a database of patients, and we're continuously watching that database of patients, and we're watching our knowledge base, and we're evaluating and looking at the patient database, able to do filtering by specific biomarkers, right? High level biomarker concepts, and being able to look at multiple biomarkers at the same time. And what that capability allows us to do is to do research. We get a cohort of patients and then do some data modeling or data science on top of that. And then it's also can be used in the clinical setting, where one of the real challenges in our field is that 5 to 10% of patients get on clinical trials. Well, we want that number to be a lot higher because we think that will help more cancer patients. But one of the challenges is that matching patients to trials is very complicated as it is. And then when you add in the precision oncology part of it with the biomarkers and things like that, our software can really help with narrowing down and trying to do the matchmaking, right? It's basically like a matchmaking service where you have patients in trials. So what our system can do is do alerts, meaning every night we're looking at the patients that have new reports or maybe new patients to the system. And then we're also looking at the new data that's coming, the new knowledge that's coming in. So for new trials that have opened up or new therapies that are available, our system can be configured to send that email to a clinical trial navigator. These are the folks that put people on clinical trials, or to treating physicians to say, hey, there's a new option for your patient that wasn't available last week. And here's what you should know about it. And so that alerting capability is really going to be critical to getting more folks on the right treatment and on the right trials. And really our knowledge graph helps enable that whole thing.
This is wonderful. Ian, you've really brought to life for us how artificial intelligence and data science can be used for good, in this case, to help treat cancer patients. Before we let you go, I just want to ask a quick question about, for our listeners today, you have, it appears, computer science training. You also mentioned there are PhDs on staff at your company. What are the various skills needed to do the work that, GenomOncology does in this very rich environment?
Right.
How would a listener develop their own pathway into a career like yours? Are there multiple pathways?
Oh, there's definitely multiple pathways. Going in high school, I got a C in biology. It was my only C in high school, and so I was not a biologist by birthright, I guess is the point. And so I've learned a lot over the last 10 years, almost 12 years, 11 years now, around DNA and exons and introns and all these other things. And it's really just been, a passion project and good mentors to help me learn these things. And so if you're a software developer and this is interesting to you, certainly reach out. And then as far as other folks that are in the field, that's, these are either, medically trained people, but then also researchers. So we have a lot of folks on our team that, have a PhD or, a master's degree in biomedical engineering. And then we also have biologists that are on our team that have PhDs. And basically what the skill is, beyond just tenacity, because tenacity, I think, is the number one skill everyone can have, is the ability to read complex information and then figure out how to break that down and organize it and structure it. And so we use, and that's what that team can do. They can read these complex eligibility criteria for clinical trials and therapies, understand the ambiguity that's being communicated, because honestly, these things are written by humans and they're not always clear. And then with that background and context, they're able to then turn those into rules in our database, and they're curating that in our system. And then we also have other folks that are then helping us design the software, right? So they're not necessarily software developers themselves, but they understand enough about the process to help us design things.
Cool. So they could take some computer science, they could come from the bio side, they can maybe even be a psychologist who decides they want to go into this space.
Exactly right.
That's very cool.
Yep.
Well, this has just been amazing. It's so nice to meet you. We really appreciate you sharing your knowledge, your insight, your background, and the ability for people to connect with you, to our broader community. At the Northeast Big Data Innovation Hub, we have over 8,300 people that we communicate with around the planet that are members of our hub. We have over 4,000 students in our National Student Data Corps, which is really international. And so they'll all get a chance to see this. So thank you very much for your time today. We really appreciate it. And we'll chat soon.
Great. Thank you so much.
You have a great day.
Thanks, you too.